In terms of neurotransmission, opioid receptors are coupled to different effectors in the cell, in the neuron. And so several things happen when an opioid binds to its receptor. First, these opioid receptors will inhibit calcium channels. So these are ion channels that are very important for neurotransmitter release. Mm-hmm. At the synaptic terminal, when neurons fire action potential, there's going to be an increase in intracellular calcium. And this signal will cause the fusion of synaptic vesicles with the plasma membrane. And so then the neurotransmitters that are inside the synaptic vesicles will be released onto the next cell to sort of activate or inhibit it. And so what opioid receptors do when they're activated is that they're going to diminish the ability of these calcium channels to let calcium in. So in the presence of an opioid, you have less calcium flowing in the synaptic terminal, less transmitter release. So this neuron is less able to communicate pain information to the next neuron in the circuit. Mm-hmm. So that's one effect. And we start the test by putting them on the hot side. So they have no choice. They have to find a solution to their pain problem. And so the treatment that they find is to go in the other chamber.
Mm-hmm. And so you can then directly measure and quantify how much they're expecting the other side to be hot, how much they don't like it, by how much time they spend in the other chamber.
Exactly. And so they're really good at this. After just 1 day, or certainly after 3 days, you see that they spend almost all their time in a chamber that's not hot.
And so then you can put such animals into the chambers like you just described a few moments ago, and you can watch to see what kind of activity these neurons display as the animals are doing this pain-related learning.
Exactly. So there are other ways to record activity of neurons, but sometimes you have to anesthetize the animals, or there are other aspects of the methodology that do not really permit the type of things that we want to do, which is in molecularly defined types of neurons in a freely behaving animal, what happens when they perform the task.
Yeah. So a strength of this way of recording is that you might not get signals that are as precise or temporally precise as say, an electrode, if you're measuring with electrodes, but you have the advantage that you can record from molecularly defined groups of neurons, particular parts of the brain, and the animals can walk around and behave just as they do in normal conditions.
Exactly. And so when we use that technique in our placebo analgesia task, what we found is that as mice are learning the task, that they're moving from chamber on the left to chamber on the right, from heat to comfort, just before they cross and they get on the other side, these cells in the cingulate cortex light up.
Well, so if you have severe enough chronic pain and you can't help it through drugs or other means, it's almost like epilepsy. Like there's severe forms of epilepsy, which, you just can't treat them other ways. So they end up going in and just slicing part of the brain. And that's what actually helps.
Yeah. So that's, cutting out the axons of these cells and preventing them from communicating this, affective signal. And the cerebellum in, pain neurobiology has always been a little mysterious. It's not easy to study. It's much more studied in the movement field, where we know it's essential for motor coordination and motor learning. In the pain field, it's often seen as activated in human imaging studies. But there's this issue that, of course, when you're presented with a noxious stimulus, you withdraw from it.
You want to move away.
Always involves movement, right?
Yeah.
And so perhaps this signal in cerebellum that is seen during these experimental studies of pain is due to movement, or even just to the intent to move. What we find in our study is that the part of the cerebellum to which the Ponty nucleus that receives this expectation signal from the cingulate cortex project in cerebellum is not the motor areas. There are particular areas of the cerebellum that are more related to cognition. And this is where this signal is going.